My name is Eric Lau, and I'm an assistant professor in the Department of Tumor Biology at Moffitt Cancer Center. And today I'm going to be telling you about a novel approach that we're taking to increase tumor-infiltrating lymphocytes in melanoma. Current immunotherapies are limited by a number of determining factors, chief among which are insufficient tumor-infiltrating lymphocytes, or TILs. This can lead to what we observe commonly as immune cold tumors. Now, a number of efforts have been undertaken to try to overcome this problem, including the combination of immunotherapies such as anti-PD-1 and anti-CTLA-4, or the combination of immunotherapies with other types of therapies such as chemotherapy and radiation. These efforts have led generally to limited enhancement, but are associated with increased adverse events and comorbidities. What I'm going to tell you about today is a completely novel approach using the administration of L-fucose as a means to boost baseline TILs before immunotherapy or TIL harvest and/or during TIL expansion. What I'm also going to tell you about is a new fucosylated MHC protein that we propose can be used as a novel immunotherapy stratification or predictive marker. So the rationale for fucose is that a number of years ago, we discovered that fucose usage is reduced in melanoma, and that simply by increasing fucose levels, we could actually suppress tumor growth by a good 40 to 50%. So in mouse models of melanoma, what we observed was that regardless of mutational background, the administration of dietary fucose to these tumor-bearing mice consistently suppressed tumor growth by about 40 to 50%. And this tumor suppression required the immune system because repeating the exact same experiment in immunodeficient mice did not result in the same tumor suppression. So we took a look at the TIL profiles in these mice whose tumors were suppressed by fucose, and what we observed was that, again, regardless of mutational background of melanoma, that we saw significant induction of TIL, anywhere from 10 to 50-fold. So we've spent the last 4 to 5 years trying to determine the underlying mechanism, and what we have discovered is twofold. First, we discovered that a class 2 MHC protein expressed on melanoma cells is fucosylated. And as we know, MHC2 proteins interact and engage with the CD4 T cell receptor complex. And what we found is that the fucosylation of this MHC2 is absolutely required for the tumor suppression that we see, because if you look at the tumor curves on the left of the bottom of the screen, if we have tumors that express wild-type MHC2, the addition of fucose results in a robust suppression of the tumor. However, when we have tumors that are reconstituted with fucosylation-deficient MHC2 mutants, the addition of fucose now no longer suppresses the tumor, indicating that the fucosylation of MHC2 is absolutely required for fucose-triggered tumor suppression. Now, knowing that MHC2 engages the CD4 T cell receptor complex, we naturally looked at CD4 T cell function in this suppressive mechanism. And what we observed was that complete depletion of CD4 T cells from mice prior to tumor engraftment completely abrogated fucose-triggered tumor suppression, as you can see by the tumor curves up on the right. Interestingly, this also completely abrogated the induction of other classical anti-tumor TILs such as NK cells, dendritic cells, and CD8 T cells. In the absence of CD4 T cells, there is no induction of these infiltrating lymphocytes by fucose. So we were keen to determine whether or not fucose might be synergistic with immune checkpoint blockade agents such as anti-PD-1. And so in an extensive mouse model, what we observed was that in comparison to either fucose alone, which partially suppressed tumors, or anti-PD-1 alone, which suppressed tumors and then resulted in relapse about 45 days later, the combination of PD-1 plus fucose together significantly suppressed tumors to the point where we didn't see any regrowth by the end of the experiment. So in summary, hopefully what I've managed to convince you of is that L-fucose represents a potential TIL-enhancing agent. We observe that L-fucose suppresses melanoma tumors and that it does so in an MHC2 fucosylation and CD4 T cell mediated way. We've observed consistent TIL inductions between 10 to 50-fold depending on the model. And so clearly these effects have implications for enhancing TIL expansion for adoptive cell transfer therapy. There are implications for enhancing immune checkpoint blockade, as we've shown in our initial mouse model. And in addition, we've also observed in our preliminary assessment of healthy human T cells that fucose can also enhance T cell activity, suggesting that it could be used during or after expansion of TIL. Now, I mentioned in the beginning that we have also developed a new predictive or stratification marker for immunotherapy, which is this fucosylated MHC2 protein. We have developed a new platform to visualize for the first time glycosylated, or in this case, fucosylated proteins. So we have optimized it both for cells, and as you can see on the bottom of your screen, we can have cells on cover slips, which we have been able to now specifically stain for the fucosylated species of MHC2. We have also optimized this now for fixed patient tissue biopsy specimens. And so on the bottom of your screen, you will also see in the green signal that that's fucosylated MHC2 on whole tumor tissue. So we propose that this method can be used to help stratify patients for different types of immunotherapy, as well as to help predict responsiveness and outcomes. And with that, I would be happy to answer any questions if you contact us via the information at the bottom of your screen.